Hello students, welcome to EPG Pathsala. I am Dr. Ranjit Singh from Environmental and Industrial Biotechnology Division, the Energy and Resources Institute, TERI, New Delhi. Today we are going to talk on the module General Principles of the Laboratory and Biosafety under the paper Environmental Biotechnology. In this module, we will be learn about the good laboratory practices, objective of GLP principles, and about biosafety guidelines. So what is a good laboratory practices? By definition, good laboratory practices is a quality systems which is concerned with the organizational processes and the conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, archived, and reported. GLP is a formal regulation created by the FDA, United States Food and Drug Administration in 1978. GLP embodies a set of principles that provide a framework within which laboratory studies are planned, performed, monitored, and archived and reported. It's a quality system of management control for research laboratories and organizations to try to ensure the uniformity, consistency, reliability, reproducibility, quality, and integrity of chemical, including pharmaceuticals, non-clinical safety test from physiochemical properties through acute to chronic toxicity test. So now we will understand why GLP were created. So this creation of GLP leads to in 1970, where numerous cases of laboratory malpractice, fraud were observed in the USA laboratories and other state organizations. They discovered that a lot of fraudulent activity and lot of poor lab practices were going on in these laboratories. Although these were originated in USA, it has worldwide impact, and this malpractices became a public safety issue. The example of some of these poor lab practices were found where the lab equipments have not been calibrated to a standard form, therefore giving wrong measurements, incorrect or inaccurate accounts of the actual lab study, and the inadequate test systems were observed. The famous example was of an industrial biotest laboratory. In this laboratory, the most notable case were observed where thousands of safety tests for chemical manufacturers were falsely claimed to have been performed or were so of poor quality. The term GLP was first used in New Zealand in 1972. The regulation of GLP becomes effective from June 1979, where assure the quality and integrity of safety non-clinical laboratory studies. GLP helps assure regulatory authorities that the data submitted are true. In 1981, an organization named OECD, which stands for Organization for Economic Cooperation and Development, produced GLP principles that are international standard. Moreover, WHO adopted the revised OECD principles of GLP as its official guidance for non-clinical safety testing. Objective of GLPs. GLP makes sure that the data submitted are a true reflection of the results that are obtained during the study. It promotes international acceptance of the tests and the laboratory practices. GLP is to certify that every step of the analysis is valid or not. It assures the quality and integrity of data submitted to FDA in support of the safety of regulated products and makes sure that data is traceable at any time. GLP also makes sure that not to indulge in any fraud activity by labs, and GLP have heavy emphasis on data record and specimen and retention of all the data. Now, what are the basic elements of GLP? So GLP consists of personnels, where personnels could be sponsors of the research programs, managements involved in the GLP regulations, study director, and quality assurance. The other elements is facility. The facility includes laboratory operation, animal care unit, equipments, reagents, and storage area. The third one is documents. The documents could be standard operating protocols, reports, or archiving of all the details of the documents. The last one is test and control articles. It includes characterization of the test, handling, and storage of the lab materials. So GLP. So this is the overview of GLP involved in from sample processing to the result analysis. Whenever the samples come in the laboratory, it goes the different laboratory processes, and then the results obtained were analyzed and documented. Let's consider it as an example of the drug company. When drug company gives any project to the laboratories, then in that laboratory GLP principles were applied and accessed. Out of this GLP, in the laboratory data were generated, and this data goes to the government and company, which are the sponsoring organizations. In this company, the hazard assessment were accessed, and after this assessment, the international trade were formulated and executed. Good laboratory practices principles. There are various principles of a good laboratory practices. The first one is test facility organization and personnel's quality assurance programs is facility, apparatus, materials and reagents, test systems, test and reference substances, standard operating procedures, performance of the study, reporting of study results, storage and retention of records and materials. So first we will discuss about test facility organizations and personnel. This includes test facility management's responsibility, study director's responsibility, principal investigator's responsibility, and finally study personnel's responsibility. So first we will discuss about test facility management's responsibility. The responsibility of the management in the good laboratory practices includes the facility should have sufficient number of qualified personnel, it have appropriate facilities, equipments and materials, and are available for the timely and proper conduct of the study. The management also ensures the maintenance of a record of the qualifications, training and experience. It also keeps records of the job description of each professional and technical individuals. They have also the responsibility of documented approval of the study plan and SOPs by the study director. So what are the study director's responsibility? The responsibility of study directors includes approval of the study plan, technical conduct of safety studies, raw data generated are fully documented and recorded, the computerized systems used in the study have been validated, the availability of SOPs to the working personnel's interpretation, analysis and documentation of all the results, reporting of the results, deviations must be acknowledged, and the record and data are archived properly. If any amendments to the study plan by date it should be signatured properly. They ensure that after completion of the study plan and the final report of raw data and supporting materials are archived properly. They sign with and date the final report to indicate acceptance of the responsibilities for the validity of the data. The third one is principal investigator's responsibility. Here, the principal investigator ensures that the delegated phase of the study are conducted in accordance with the applicable principle of good laboratory practices and other safety measures. The fourth one is study personnel's responsibility. The personnel involved in the study should have the knowledge of the GLP principles. They have access to the study plan and appropriate SOPs. The personnel must comply with the instructions of the SOPs. They record all the raw data generated. They are responsible for the quality of their raw data. exercise health precautions to minimize risk, and they ensure the integrity of the study. The second principles of GLP is Quality Assurance Program. In this program, designated individuals are members of the quality assessment team. They are directly responsible to the management. QA members should not be involved in the conduct of the study, as they are only for the quality assessment. What are the other responsibilities of the quality assessment personnel? They have the access to the updated study plans and SOPs. They keep all the documented verification of the compliance of study plan to the GLP principles. They inspect to determine compliance of the study with GLP principles. Generally, there are three types of inspections. The first is study-based inspections. The second is facility-based inspections, and the third one is process-based inspections. These inspections of the final report happens for the accurate and full description. Report the inspection results to the management. The third one is facilities. So first we will talk about test system facilities. The test system facility is designed for the test facility which should provide an adequate degree of separation of the different activities to ensure the proper conduct of each study. It must have sufficient number of rooms or areas, assure the isolation of test systems and the isolation of individual projects involving substances or organisms known to be or suspected of being biohazardous. There should be storage rooms or areas as needed for supplies and equipments. Separate animal care unit must be there. And the area should be available for the diagnosis, treatment and control of disease in order to ensure that there is no unacceptable degree of deterioration of the test systems. The test and control products maintained in a secure area. They must have a separate suite, a specimen and data storage facilities, and it must have appropriately qualified personnel. Archive facilities. The archive facilities should be provided for the secure storage and retrieval of study plan, raw data, final reports, sample of test items and specimens. These archive designs and archive conditions should protect contents from untimely deteriorations and other adverse conditions. The last one is waste disposal mechanism. As the handling and disposal of waste should be carried out in such a way as not to jeopardize the integrity of study. This includes provision for appropriate collections, storage and disposal facilities, and decontamination and transportation procedures. The apparatus, materials and reagents. The apparatus must be of appropriate design and adequate capacities, including validated computerized systems used for the generation, storage and retrieval of data and for controlling environmental factors relevant to the study. The apparatus used in a study should be periodically inspected, cleaned, maintained and calibrated according to the SOPs. The apparatus and materials used in a particular study should not interfere adversely with the test systems. And the chemicals, reagent, solutions should be labeled to indicate identity with concentrations if available, expiry date and specific storage instructions. The information concerning source, preparation date and stability should be available also. The fifth one is test systems. The test system includes physical and chemical test systems and biological test systems. In physical and chemical test systems, there are appropriate designs and adequate capacity of the apparatus involved. Integrity and physical chemical test systems. The biological test system includes proper conditioning for storage, housing, handling and care. They also include isolation of newly received animals and plant test systems, humanly destruction of inappropriate test systems, records of source, data of arrival and arrival conditions of the test systems. Biological test system also involves proper identification and acclimatization of test systems, whether it is plant or animals, in their container or whenever they are removed for the laboratory testings. Cleaning and sanitization of the containers on regular basis, documentation of pest control agents to be used. The sixth principle is test and reference items. Test and reference items include receipts, handling, sampling and storage, date of receipts, expiry date, quantities received and used in studies all should be labeled properly on the containers, handling, sampling and storage procedures, identification information on storage container, then the characterization of the tests. Identification of each batch of test and reference items should be in batch number, purity, composition, concentration, everything should be mentioned for the characterization. The verification of identity of the test systems, known stability of test and reference items, stability of the test items in its vehicle or container. Finally, experiments to determine stability in tank mixes used in the studies. The seventh principle is standard operating procedures, or in short called SOPs. SOPs are documents procedures which describe how to perform tests or activities normally not specified in detail in study plans or test guidelines. These SOPs are approved to ensure the quality and integrity of the laboratory data. They are derived from SOPs should be acknowledged by the study director. The SOPs are prepared for test and reference items. It includes routine inspection, cleaning, maintenance, testing and calibration. Actions to be taken in response to equipment failure. The SOPs are for keeping records, reporting, storage, mixing and retrieval of data. Definition of raw data, analytical methods, keeping records, reporting, storage, mixing and retrieval of data, statistical procedures for data evaluation. Practitioners in a particular field may adopt certain standards which are deemed acceptable within their field. Regulatory agencies often describe acceptable statistical procedures. The scope of the SOPs are applied to the non-clinical safety testing of test items that contained in pharmaceuticals product, pesticides products, cosmetics products, veterinary, as well as food additives and industrial chemicals. The principles of SOPs are related to work conducted in the laboratory, greenhouse or in the field. The next principle is the performance of the study and the reporting of the study results. Performance of the study are done to prepare the study plan. These are the written plan verified for GLP compliance and approved by the study director and by the management of the test facility. The content of the study plan includes identification of the study, the title, nature and purpose of the study, test items identify, reference item used, etc. It also includes records. For example, name and address of the sponsor, test facility, study director. It also have dates, which is the approval date of the study plan, estimated starting and completion date. It should also include references to the test methods. The reporting of study results includes final report of each study with sign and date, approval by the study director, information on sponsor and test facility. This report includes the quality assurance program statement, description of materials and test methods, results and storage, which include storage of sample storage, reference items, raw data, final report, etc. The 10th principle is storage and retention of records and materials. So retention, what to retain in archives? So there is a very selective criteria for this. Records should include the study plan, raw data, samples. It also includes inspection data and master schedules, SOPs of all the lab testing procedures, maintenance and calibration of all the data. If any study material is disposed of before expiry, the reasons should be justified and documented. It also includes index of material retained and environmental assessment of health and safety monitoring records. This all should be stored and retained in the proper documentation forms. So what good laboratory must contain? A good laboratory must have a clear and well ventilated area that should be free from smoke, smell and dust. The good laboratory should ensure good ventilation, proper illumination and preference given to natural light, air conditioned in the laboratory with humidity control. A good laboratory should have enough space for measuring and testing instruments. It must have proper arrangement of testing. It should take care of all safety points including proper fire safety. It should avoid uncleanable spots in floor, walls and ceiling. A good laboratory establishes proper area for storage of incoming samples as well as test completed samples. It also provides sample collection place as well as parking and disposal of tested samples. So what are the benefits of good laboratory practices? So if we follow the GLP, it can be very advantageous for our research work and our other laboratory process equipments and other and laboratory colleagues, as it will give better image of organizations as a quality producer in global market. A good GLP will provide hot tips on analysis of data as well as measure uncertainty and perfect record keeping. It will provide guidelines for doing testing and measurements in detail with all the record documented, and that can be traceable anytime. It will provide guidelines and better control of maintenance of instruments, environment control, preservation of test records, etc. Maintenance and calibration of equipment. So equipment shall be adequately inspected, cleaned and maintained on regular basis. Equipment used for the assessment of data shall be tested, calibrated and standardized. The scales and balances should be calibrated at regular intervals. No, for reagent and material certification. This policy for this certification is to ensure that reagents used are specified in the standard operating procedures. The reagent and materials are purchasing and testing should be handled by a quality assurance program. So what are the requirements of these facilities? The reagents and solutions shall be labeled properly. Deteriorated or outdated reagents and solutions shall not be used. It includes date opened, date received, etc. The storage under ambient temperature or unless specified on the material safety data sheets. The expiration date should be properly labeled on the reagents. What will happen if workplace doesn't comply with FDA GLP standards? So this non-compliance of GLP standards could disqualify the facilities, as before a workplace can experience the consequences of non-compliance, an explanation of disqualification will be required. The FDA states the purpose of disqualification as the exclusion of a testing facility from completing laboratory studies or starting any new studies due to not following the standards of compliance set by the Good Laboratory Practices Manual. The possible violations will include falsifying information for permit, registration or any required records, falsifying information related to testing, protocols, ingredients, observations, data, equipments, etc. Failure to prepare, retain or submit written records that is required by the law. So what are the consequences of non-compliance of the GLP? The FDA states the following consequences of non-compliance. For example, the commissioner will send a written proposal of disqualification to the testing facilities. A regulatory hearing on the disqualification will be scheduled. Then, if the commissioner finds that after the hearing the facility has compiled then a written statement with an explanation of termination of disqualification will be sent to the facility management. Thus, if it can be shown that such disqualification did not affect the integrity and outcome of the study itself or did not occur at all, then the study may be reinstated at the will of the commissioner. Upon disqualifications, if the commissioner find that the facility was non-compliant on any of the grounds after the hearing, then a final order of non-compliance will be sent to the facility with explanations. If a testing facility has been disqualified, then any studies done before of or after the disqualification will need to be determined as essential to decisions which is acceptable or not. If the study is determined unacceptable, then the facility itself may need to show that the study was not affected by the non-compliance that led to the disqualification. Once finally disqualified, the facility may not receive or may not be considered for a research or marketing permit and the study is rejected. Also, the commissioner may notify the public and all interested persons, including other federal agencies the facility may have contacted earlier. The FDA may ask the other agencies to consider whether to support the facility or not under the disqualifications. The FDA may turn it over to the federal or state or local law enforcement for further actions. The facility's sponsor may terminate or suspend the facility from doing any non-clinical studies for a permit. Also, the sponsor is required to notify the FDA in writing within 15 working days that the facility is to be suspended or terminated and why. Reinstatement of a disqualified facility. The testing facility may be reinstated as acceptable non-clinical study to be turned into the FDA if the commissioner can be certain that future studies will be conducted in compliance with the GLP standards and that any current studies' integrity have not been severely harmed by the disqualification. Moreover, the disqualified facility will be required to put in writing to the commissioner reason why it should be reinstated and any actions the facility will take or have taken to assure any disqualification problems will not happen again. Then the commissioner will inspect the facility and determine if it shall be reinstated or not. If it is reinstated, the commissioner is required to notify all persons that were notified of the disqualification, including the facility itself and the study director and other management personnel. So what are the general laboratory biosafety? Why does this biosafety matter to us? This biosafety is a safe working prospects. It is safe for you, for other lab workers, for cleaners, for visitors, and most importantly for your work. So what does the law say about the biosafety? The first important point is Health Safety at Work Act of 1974, which says you must work safely, you must not endanger others, lab colleague, you must not misuse safety equipment. There is a provision of penalty in this law which says up to 2 years in prison or an unlimited fine. The Management of Health and Safety at Work Regulations 1999. The Control of Substances Hazardous to Health Regulations 2004. Hence, you must perform risk assessment always. So how do a risk assessment happens? Or how we measure the risk assessment? So you have to determine hazard and evaluate the risk. For example, you use all relevant available data, determine controls needed to minimize those risks, document the assessment so that it will be helpful for the others, agree it with your supervisor, use those control measures for the future works and experiments, and get a specific training on how to do this risk assessment in your laboratory. So what are the control measures? So here are some rules of control measures in the order of preference. The first one is use a less risky substance. Use a safer form of that substance, for example, solution instead of a powder. You also totally enclose the process. For example, a glove box, you should partially enclose the process. For example, with a fume cupboard, you must ensure good general ventilation, safe systems of work, reduce exposure times, increase distance, reduce volumes. Personal protective equipment should always be wear, such as last resort for primary protection. So how we should protect ourselves? So wear the clothing and protective wear which is identified in your risk assessment. Laboratory coat must be kept fastened always whenever you are in the laboratory. Don't wear sandals or open shoes. Long hair must be tied back. And there are many different types of protective gloves. Use the correct one for the job you will be doing. Remember always that you need to select chemical protection gloves according to the materials or substances with which you will be working. Remove your gloves before using instruments, telephone, and leaving the laboratory always. So what are the general hazards in the laboratory? The general hazards are fire, breakage of glassware, sharps, spillages, pressure equipment and gas cylinders, extremes of heat and cold, chemical hazards, biological hazards and radiations, and there are many more other hazards. The first one hazards is fire. So how we should avoid fires? Fires are from flammable substances, so use minimum quantity of the flammable substances. Store these substances in a special storage cabinet and use temperature controlled heating sources. For example, water bath rather than hot plate or Bunsen burner. So how should we minimize the fire damage? For that, you should always make sure corridor fire doors and laboratory doors are kept shut at all the time. You should take in the fire safety measures. Make sure that you know what to do if you have a fire, if you hear a fire alarms, and if you are a member of staff, you must attend fire training annually. Postgraduates should also seriously consider doing so, as the other labs members are doing. The next hazard is glassware. Use correct techniques for the insertions of tubing in glasswares. Never use glassware under pressure or vacuum unless it is designed for the job and suitably sealed. Dispose of chipped or broken glassware. It is a risk to you and others. Always dispose of broken glass in a glass bin or sharps bin and not in general waste bin. If spillage hazardous happens, then what we should do? First, clean up spillage promptly. You will already have determined how to do this as part of your risk assessment, and dispose of any hazardous material as toxic waste. As it is generally considered as messy workers are always usually poor workers. The next hazard is gas cylinder. As never used without formal training, you should minimize the number in a laboratory. Store externally whenever possible. And these cylinders are heavy and can cause serious damage if they are fallen on your foot. Ensure that they are chained when in use, and cylinders move only with a cylinder trolley. Use regulator and control equipments for the safety control of the cylinders. Cryogenics. As the liquid gases are extremely cold and can cause burns. Also, these liquid gases evaporate and may cause asphyxiations. If you need to take cryogens in a lift, there are special procedures to follow. Speak to a supervisor or a senior member of technical staff. And you must have special training to use them in a very safety manner. Electrical equipment. Always do a visual check on electrical equipment before use, looking for obvious wear or defect, as all electrical equipment must have a current pad test stickers, and never use defective equipment always. The general tidiness in the lab. Keep your workplace tidy, clear of the waste, deal with washing up and put things away as you finish with them. And make sure everything is safe before you leave things unattended. A tidy laboratory avoids accidents to everyone. Laboratory equipments. You should never use any laboratory equipment unless you are trained and have been authorized to do so. As well as you will get injured yourself and you may cause very costly damage to the laboratory. and the managements. First aid measures. All laboratory workers should undergo simple first aid training. For all chemical splashes, wash your hand with plenty of water for approximately 10 minutes. Control bleeding with direct pressures and avoid any foreign bodies such as glass. Report all these accidents to your supervisor or departmental safety officers. More importantly, protect yourself and your health. If you have an allergy to lab materials or suffer formal or medical condition which may affect you in the laboratory, for example, if you have diabetes or epilepsy, ensure that your supervisor must know about your health condition. The next hazard material is a waste material. Part of your risk assessment will be to determine how to dispose of your laboratory waste materials in very safely. This can be done through solvent and while must be segregated first into the correct waste bottles or drum. Then your department will help you determine what to do with chemical or the biological materials waste. You should not put materials down the drain or in with normal waste unless authorized to do so. You should follow all the instructions of the laboratory safety and waste disposal guidelines measurements. Now we come to the summary part. So students, let's now summarize what we have learned in this module. We have learned that GLP is an FDA regulation which is accepted and approved as international standard by OECD to avoid the fraud activities of the testing laboratories for pesticides, pharmaceuticals, food additives, dyes. It is essential to save the human and environmental health and also to correct the good international trade and establish good relationships among the countries. It gives better image of company as a quality producers in global market, which provides hot tips on analysis of data as well as measures uncertainty and perfect record keeping and guidelines for doing testing and measurements in details. Finally, GLP provide guidelines and better control for maintenance of instruments, environment control, preservation of test records, etc. Good laboratory practice regulations are applied to non-clinical safety of study items contained in pharmaceuticals products, cosmetic products, veterinary drugs, devices as well as food additives. The purpose of testing these items is to obtain information on their safety with respect to human health and environment. GLP is also required for registration purpose and licensing of pharmaceuticals, pesticides, food additives, veterinary drug products and some bioproducts. Thank you very much.